So today I'm going to talk about the technique that we've been using called microcrystal electron diffraction or micro-ED. It's a technique that I developed during my postdoc, and now since I've moved to my own lab about 4 years ago, we're continuing to develop this both in my lab and in collaboration with other labs around the country. So first off, these are introduction slide. I don't need to spend a ton of time in an audience like this explaining that X-ray crystallography has been enormously successful, but there are some bottlenecks, which is mostly that you need to grow large crystals, relatively large crystals, for structure determination. And oftentimes, as I'm sure many in the audience have experienced, it's not always easy to grow big crystals that diffract really well. So when you have crystals that look like this, which are obviously big, beautiful crystals, no problem, everything's great. But what happens if you get something more like this, which is just a shower of tiny little things, and no matter how hard you try, you can't seem to ever get them bigger. So there's been a need to develop new methods to sort of try to use these small, tiny little crystals to determine structures from these little crystals. And a lot of different techniques have come out, right? Of course, synchrotrons are getting bigger, better. They're being able to shoot smaller things. You've got the free electron lasers, which are also sort of probing these small crystals. But we sort of took a different approach and said, well, instead of X-rays, can we use electrons? So we're going to use electron diffraction to sort of look at these small crystals. The main point is that electrons interact with matter much stronger than X-rays. They deposit less damaging radiation compared to X-rays. So that means you can get structural data from smaller and smaller crystals. So this technique of micro-ED is within sort of the field of cryo-EM. Now we've seen a lot of talks in most of these, especially electron tomography and single particle cryo-EM. So I'm not going to spend a ton of time on these, but these are the ones that people are more familiar with. These are imaging-based techniques. You also have sort of crystal-based techniques. Two-dimensional electron crystallography, which also uses imaging or diffraction. It's actually sort of one of the first sort of high-resolution techniques within cryo-EM with sort of Richard Henderson and Nigel Enwin. So these use two-dimensional crystals, whereas this sort of microcrystal electron diffraction uses thin three-dimensional crystals. So it's kind of within this sort of broad field of cryo-EM, which in general, like, a while ago you could say this would go in order of, like, being more biologically relevant, but then higher resolution this way, but now single particles kind of encroaching on the crystallography world, which is great. But not a lot of people within cryo-EM necessarily are familiar with the diffraction-based technique. So I have this slide up here just to kind of orient everybody, if they're familiar with cryo-TEM, that your cryo-TEM can also do diffraction, right? Especially if you have an FEI microscope, or I guess Thermo Fisher now. There's a button on it that says diffraction, and you push that button, and you can collect diffraction patterns instead of imaging. I won't go into a ton of it, but it changes the lenses, and basically, when you're imaging here, right, here's your sample, and you're basically projecting the image plane down the detector, whereas in diffraction, you're projecting this diffraction plane onto your detector. You do a few different things, right? In imaging, you have your objective aperture in, you take your selected area aperture out, you do the opposite diffraction, you keep your objective aperture out, and put your selected area aperture in. But the point is, this is the exact same instrument that you use for imaging, you can also use for diffraction, which makes this technique really nice because you don't need to buy a completely different instrument than what many institutions already have. So this is just kind of orient everybody. All right, so this slide was more for the cryo-EM crowd. So within this technique, you've got elements of cryo-EM, you've got elements of diffraction. So usually the cryo-EM people are comfortable with that slide, and the X-ray diffraction people are more comfortable with this slide, which is, okay, what happens when you have a diffraction pattern? Well, they look different. You have X-ray diffraction right here. This is lysozyme, which I'm sure many of us have shot before. You get these rings, right? And you're sampling, if you pretend these are spots in reciprocal space, you've got H, K, or you've got H, L, and then you've got the other dimension kind of coming out at you. This is the Ewald sphere, which is where Bragg's law is satisfied, and all, that's where the spots will show up in the detector. You're sampling three-dimensional space. In electron diffraction, because the relativistic wavelength is so incredibly small, your Ewald sphere is essentially flat, which means you're doing a really good job of sampling in 2 dimensions, but a terrible job getting 3-dimensional information. And so this was the big problem, is crystal orientation can't be defined by a single diffraction pattern for electron diffraction. Now in 2D electron diffraction, for 2D crystals, that wasn't a problem because they always sort of orient with the c-axis pointing straight up, so you already kind of know what that third dimension is, and you can go from there. But for 3D crystals that might land different ways in the grid, it's not immediately obvious how to index that. So that was kind of one of the big hurdles with using thin 3D crystals for structure determination. I just want to zoom this in because I really like this picture. This is also lysozyme, electron diffraction pattern, and it's right along the 001 sort of plane there. And you can actually see the symmetry. You can see here's your A and your B. You can see the systematic absences here. You can see the fourfold symmetry. It's really cool to see these, because the Ewald sphere is so flat, you basically you're looking at the planes themselves. And so you can see the systematic absences right there. It's just a really pretty picture, I can show that. So sample preparation, real quick before we move on. How do we make samples and get them into the CryoEM? And the workflow is pretty much identical to single particle CryoEM. You take a slurry of protein microcrystals or any sort of microcrystals, you put them on a wholly carbon TEM grid, you vitrify it, you blot it, you vitrify it, liquid ethane, and then you load it in the microscope, you look around for microcrystals. And when you see microcrystals, then you can see if they diffract. But very similar process to single particle CryoEM. Okay, so this is the big problem that we found is, okay, if you have a small microcrystal and you give it the dose that you would normally give a sample if you're doing a two-dimensional crystal, for example, you'd only really collect one diffraction pattern, and then the crystal would be sort of dead, and you wouldn't know how to index that crystal necessarily, and so you'd have one maybe really pretty diffraction pattern, but that's about it. So the key for this was, okay, well, if we're going to say our limit of radiation damage is somewhere around here, that means we have to collect a lot more diffraction patterns, sort of at known orientation, so we can fully sample reciprocal space in 3 dimensions. And so that was sort of the first thing we did was, okay, can we shoot this at a very, very low dose? This is way lower than we use for imaging in single particle CryoEM. And sure enough, you can get information to very high resolution even at these low doses. So what that means is we can take multiple diffraction patterns from a single crystal and sort of look at different orientations. And because if we know how every diffraction pattern is oriented relative to the previous one, we can start to extract 3-dimensional information. And so that's how we collect the data. We collect data by what's called continuous rotation data collection. So in this little cartoon diagram over here, again, this is our representation of reciprocal space. This is the electron source. Here's a crystal on the stage, and then down here is our detector. And so what we do is we tilt the crystal, and if we were to take a single shot, we sort of sample reciprocal space right here. What we do, though, is we use these fast cameras that can record continuously, and we turn the beam on and we rotate the crystal, and that allows us to sort of sample 3-dimensional space. So this camera is continuously recording, and so you're getting wedges of data, which look a lot like the wedges of data you get from an X-ray diffraction experiment. What does the data look like? So here's, again, the pattern. Instead of the lunes, you get these straight lines because the Ewald sphere is flat. And as you tilt the crystal, you can sort of see the crystal move, the spots come in and come out as you tilt the crystal. And because we know the rotation rate of the stage, and we know our integration time, we know sort of the wedge of data that we're integrating over, which makes this process really, really nice because you can just put this data into standard X-ray processing programs, and they handle the data just fine. So this was in collaboration. We worked with Andrew Leslie originally on this, and that's why you'll see we have moss film up here. But we basically take this diffraction data, we convert the TIFFs that come out of the microscope, and put them into a format where the programs think they're X-ray diffraction files, because otherwise the programs won't load them. But you trick it. We see if for moss film we had to rotate because it wanted the rotation axis to be this way, even though in the microscope it's this way. But you do these little tricks. You tell the Ewald sphere is really, really big. You tell, instead of the detector being 400 millimeters away, the detector is now 2,000 millimeters away. And other than that, the programs run pretty surprisingly well, actually. So you see here, spot finding moss film, if you're familiar with moss film. finds all the spots. It indexes, you probably can't read that, but it shows that it found 76.2, 76.2, 37 angstroms, 90 degrees, 90 degrees, 90 degrees, which is tetragonal lysozyme. You can see here are the predictions of all the spots in each frame. Falls really, really, it aligns very well. And you can integrate that into a full three-dimensional data set. Now that comes out as sort of an MPZ file. You can run that through Aimless or whatever your favorite programs are, and you get a file that looks just like an X-ray data set. And so here's an example of the results we get. This is for proteinase K. This is the density map right here. Just note it's not an electron density map, it's just the density map because electrons are seeing different things. But it looks exactly like an electron density map, and if you're interested, you can kind of look at these statistics. They're very similar to what you'd expect for an X-ray structure. There are a few things which we can talk about in the questions, or we can talk later about. Sometimes the R-frees and stuff are a little high. There's reasons for that. But other than that, it works pretty well. So that's the quick run through of how sort of microED works. And since we have worked on it, both our lab and other labs have solved quite a few structures, different proteins, these different peptides, which was a really great collaboration between our lab and David Eisenberg's lab at UCLA. Sort of served as the first real sample. They brought these amylogenic peptide fragments to our lab, and sure enough, they diffracted to like 1 angstrom, and we were able to solve a whole bunch of their structures for them. And a lot of these structures, a lot of these peptides, especially this one here, they've been trying to get diffraction data from this for, I think, almost over 5 years. They tried XFEL, they tried everything, they couldn't get it, and then we solved this structure in about an hour and a half, I think, when we finally got the electron diffraction data. So when it works really, really well. The problem is it doesn't always work really, really well, and like anything, further method development is sort of critical, and that's kind of where a lot of my research is focused now, sort of pushing the method forward. Can we make it easier, better, faster, all that kind of thing. And just to highlight sort of the need for method improvement, so this was the first structure. The first structure that we published was the first example of a three-dimensional crystal providing a full structure in an electron microscope. This one took quite a while. It took almost 5 months because we were actually hand indexing all the diffraction patterns because we weren't collecting it quite the same way. But we still got a structure about 2.93 angstroms. Just a few months later, once we started collecting the data continuously, and we worked with Andrew Leslie, we kind of used some of these programs, the exact same batch of crystals, like the same grids, now with 2.5 angstroms, just by improving the way we collected the data, the way we processed the data. Then a few years later, through more improvements of data processing and data collection, the same lysozyme crystals, these are no longer the same batch, but made the same way, now go to about 1.8 angstroms. So you can see the improvement driven just by sort of methods improvement. And in this sort of audience, I don't think I need to make, that's not shocking that methods improvement is important. But that's kind of where we're focusing on now. So there's a lot of projects in my lab where we're trying to extend microED to things like difficult samples like membrane proteins, things that are in very viscous media, because you need to block those samples very, very thin, just like in single particle. Because the nice thing about electrons is they interact with matter very strongly. The bad part about electrons is they interact with matter very strongly. So that means you have to block the sample really thin, and if you've got 40% PEG 5000 or something like that, that's not going to blot away. So we're trying to figure out new ways of handling this. And so I'd be happy to talk about that with anybody afterwards, but one of the areas that's kind of getting popular right now, and I was sort of asked to talk about, was using microED for small molecules. So this has recently got kind of popular. A couple of groups, my old lab, my old postdoc lab, and a few other labs in Europe show that you can just take raw sort of powder from some sort of organic synthesis, put it on a grid, and solve high resolution structures sort of right from the unpurified material. And so, being a structural biologist, I didn't really recognize that the organic chemistry community would find this very useful, but apparently they're getting very excited about it. So you can use microED for these small molecules, and so this is the first story. This has only been going on for a couple of months in my lab, but we're working with a group, the College University of New York. They're an organic synthesis lab. They make these small molecules for sort of organic photovoltaic kind of things, stuff that I'm not a huge expert in. But what they do is they have these large compounds, and then they assemble them into sort of super molecular compounds that have different sort of electronic properties. and they have a hard time getting the structures of a lot of these. They mostly do NMR, and sometimes NMR works, but sometimes it doesn't, and once it doesn't, they're kind of stuck. And a few of them, they've gotten big crystals, but some other ones, they haven't been able to get big crystals. And so they sent some samples to us to take a look at this, and so I'm just going to show you kind of what this process looks like. So here's was their first sort of compound. This would be compound A that they then would mix with compound B, and they'd make this new supermolecular compound. And so you can see here, it makes tons of these little crystals all over the grid. We actually collect data from things that look more like this right here, this little thing that you probably would just ignore. That's kind of what we shoot at. And you can see it gives you a diffraction pattern that looks like this. So we collected some diffraction data. I think this sample, we actually saw visible reflections to about 0.55 angstroms. But we didn't collect that high because this was sort of the first, this was actually the first data we collected on our detector. So this was collected on our Titan Cryos with the C to D detector. And you can see what the data looks like. So here's again the continuous rotation data. You can see as we're continuously rotating, the spots are coming in, coming out. We know the angular range that we're sampling over, so now we can feed this into our data processing program. So we feed this into XDS, process the data in XDS, and then use sort of the ShellX suite to then to solve the structure. So after we put this into ShellXT, we get something that looks like this. And so this is the molecule, which looks very similar to what they thought it should look like. What they're really interested though is the crystal packing. What was interesting to them was that all these 5-membered rings are all kind of stacked on top of each other. They thought there'd be a lot more sort of pie stacking, and they were finding out that it's really not. They're kind of shifted a little bit. So they were really excited about this, and then this wasn't the compound they were really searching for, but then as they dug through their files, they said, okay, well this is compound A. I looked back at some other data, and we actually saw there's actually at least one other unit cell and maybe a third unit cell on the same grid that we have data for. So we think we might actually have from one grid compound A, compound B, and then sort of the complex together. Just sort of from one grid, we're getting all of the data. We only spent about an hour and a half collecting this data, so I'm sure if we'd gone back and collected more data, we could probably have solved all 3 of them, and we're going to return to this later. So the good news is we solved the structure. The not so good news is they realized later that they actually did solve the structure of this by X-ray. You can see this down here. I didn't know this, and they actually forgot about this, but you can see it's, very, very similar. So this was actually a nice validation because we solved the structure completely blind, not knowing there was an X-ray structure, got the structure, and then found out a couple weeks later, oh, they did solve the structure. And the big difference here is, the crystal size, about 0.05 cubic microns and about 1,000 cubic microns. So I was really excited to sort of validate, okay, we're getting data, because this was the first data we collected on this microscope, so that was really exciting. Another quick story, just again to kind of show you how this works and how useful this could maybe be. Friend's lab, he's in the biochemistry department, he's a friend of mine, he does unnatural amino acids. And so we're trying to make this unnatural amino acid right here. It essentially looks like tyrosine, but with another sort of hydroxyl component. And they're trying to use this enzymatic process to make this unnatural amino acid for some other purpose. I'm not quite sure what it is. But they made something, they didn't know what it was. They took it to NMR, they got an answer, but it was kind of vague. They tried NMR again, didn't get an answer. Then they tried NMR a third time, they got an answer, but they couldn't interpret it. They took it to the NMR expert, and the NMR expert's he said, oh, you've basically got this weird thing where this beta carbon is not here, but rather it's over here. So the beta carbon's gone and replaced the hydrogen on what would normally be the amino acid sort of backbone right here. And they couldn't understand how that would happen, and so they kind of went back and forth with the NMR guy. So I said, well, why don't we just take the sample? So we took the sample, again, made a little grid of it. We're shooting this little thing right here. It's like 0.5 micron by 1 micron by a couple hundred nanometers thick. Diffracted really, really well. This is sort of the output of XDS. You can see all the predictions or the circles and all the reflections or the dark circles here. Took this data, ran it through ShellXT, just searching for all carbons, and this is what we get. And we see, sure enough, yep, we're missing the beta carbon, and it's been moved back here. And again, we collected data. It took about 30 minutes to collect the data, about 10 minutes to sort of solve the structure, and then we could kind of solve the fight between the NMR people and the synthesis people that, yeah, you did make some weird compound. So they're actually in the process of figuring out how is that possible. They think that maybe something happened where the reaction is actually happening outside of the enzyme. But here's sort of the molecule. So after taking this, we built all the, turned got rid of all the carbons here and put the right elements in. And it's actually, when we solved it, it looked like that they actually had both enantiomers were in the same unit cell. And then they did some tests and found out, yeah, sure enough, they had this sort of racemic mixture. So it was kind of a cool little project. This data we collected actually on a really old TF20. How old? Well, it still says Phillips on it, so that's how old it is. And we have a new TVIPS XF416 camera on it. So a relatively simple system. We didn't even do cryo. This is all room temperature. And we're able to sort of solve the structure pretty quick. So you don't even need a fancy Titan Krios to do a lot of this stuff. We placed a lot of these hydrogens just sort of within shell XLE. We kind of placed it in the peaks. But you can see if you remove the hydrogens and look at the map, you see there is density for a lot of these hydrogens. You can see here this hydrogen right here coming off the hydroxyl makes this hydrogen bond with the next carboxylic acid group and the next one over. And so you can see a lot of these densities for the hydrogens. We don't see all of them, but we do see some hydrogens. So again, we've continued, a lot of people are pushing this. There's a paper that just came out from a group in the Czech Republic where they did a full dynamic scattering refinement, and they can sort of pick out all the hydrogens now in the atom. They can even distinguish between carbon and nitrogen and oxygen, things like that, which is incredibly hard for electrons because the scattering difference between carbon, nitrogen, and oxygen is not as big as the scattering difference with X-rays. So people are continuing to push this, and so one day we will be able to we'll probably see all these hydrogens. So with that, I'll kind of just conclude. Hopefully it convinced you that micro-ED is capable of structure determination using very, very small crystals, and when it works really well and really quickly. So we published the first structure in 2013, and since then we kind of have been continuing to improve this, and it's continued development is critical to kind of keep pushing this forward, especially with proteins. The protein side is a lot more difficult, as with everything biological. The sample preparation is a lot harder. The small molecule and peptide stuff, though, is pretty straightforward, actually. And so I'd like to acknowledge everybody who worked on this work, everybody in my lab right here, people at ASU that kind of run the microscopes, my collaborators in New York and at UCLA, my old lab. They were in Biennial, now they're at UCLA, which is a lot closer to Arizona, so we get to visit each other more. And my funding, and with that, I'll take any questions.